Hey all you cool cats and kittens. In this video, we're going to go over the answers for April 2nd. And we are continuing to look at how DNA is replicated in eukaryotes. So we're looking at poly 1, polymerase 1. So what's the main function of poly 1? Poly 1 Pol 1 will work on the ligand strand and it will remove the RNA primers and put DNA in its place. So it's important to have 5' to 3' because that's what's cutting the primers. If you look, we're going 5' to 3', so I cut. While if I make a mistake, I need to be able to go backwards and cut 3' to 5'. So that's why it's important to have both of those exonuclease activities. So what are the structural features of the Klenow fragment? Alright, so first off, generally what does it look like? It looks like a right-handed claw. Here you have your exonuclease activity, 3' to 5' in the palm. If you're Pol 1, you also have 5' to 3' activity. In this groove where DNA binds, you have a bunch of positive amino acids, which makes sense because DNA is negatively charged, so you want positive amino acids to bind your DNA. How does the Klenow fragment know when a Watson-Crick base pair has been made and the correct nucleotide has been put in? That's because when a Watson-Crick base pair is made, you form hydrogen bonds in the amino acid side chains in the nucleotide you're trying to add. If the hydrogen bonds are not made to the side chain, then you know it's a non-Watson-Crick base pair, and so you're not going to go to the closed formation. So in the open formation, you're binding DNA, trying to figure out what nucleotide goes in there. Once you made the hydrogen bonds, you close your claws, and you add this new base to your DNA. Alright, so I forgot to add this question on here, but what are the roles of the metal ions? And so we have both magnesiums. So magnesium B is holding this nucleotide in place, helping to shield the negative charge. Magnesium A is also interacting with our NTP, but it's also adding our target and helping to stay that in place and also bridging this negative charge. What is ORI-C? ORI-C is the origin, where in prokaryotic replication, it's a sequence 245 base pair where we will start DNA replication. The first thing that binds is DnaA, binds to a AT-rich 45 base pair sequence in the ORI-C. After DnaA, DnaB will be recruited, and DnaB is a helicase. That's what's actually unzipping your DNA for you. How does DnaB make its way across the genome? That's our hand-over-hand movement that we talked about in the video, where DnaB is a hexamer. The topmost subunit of that hexamer, here's A, is going to spend some kind of NTP, so it's going to burn energy, dissociate from its neighbor, here F, move 12 residues down to associate now with B, and when it does that, we unzip 2 more nucleotides, and we do that over and over and over and over again. What prevents single-stranded DNA from forming a double-stranded DNA? Single-stranded binding protein can only bind to single-stranded DNA, so a coat single-stranded DNA, make sure that you will not form double-stranded DNA. How many copies of Pol 3 are in the replisome? That would be 2 copies, one for the leading strand, one for the lagging strand. We'll see in eukaryotic replication we have more, but in prokaryotic E. coli there's 2. And what's the trombone model? Since these 2 Pol 3s are connected by the replisome, they cannot move far apart from each other. So to be able to do both the leading strand and lagging strand at the same time, the lagging strand kind of has to bend over on itself so it can interact with the Pol 3. That's the trombone model. What's the beta clamp structure? How does that relate to its function? So the clamp is a giant circle with a hole in the middle. DNA goes through that hole, and the residues inside the hole lightly associates the DNA, so allows the clamp to stay associated with DNA, but not that strong, so it can move around. And the function of the beta clamp is simply to allow poly3 to have high processivity. So by staying on the DNA and moving around, poly3 is attached and it'll stay on the DNA. What is the structure and function of the replisome? So the replisome, the function is to synthesize new DNA for the leading and lagging strand. Structurally, this is how it does it. So you have a clamp loader. The clamp loader is connected both to the polymerase, both poly3s, and it's also connected to a helicase and a primase. So everything is in close proximity. The helicase is unwinding DNA, and it's also adding RNA primers for new Okazaki fragments. So every time you want to synthesize an Okazaki fragment, you need to have a beta clamp there so DNA polymerase can hang on. So the clamp loader will load a new beta clamp for every Okazaki fragment, and then DNA polymerase will be added on and be associated with the beta clamp. It's attracted by the beta clamp, so it'll go and find it. Then the beta clamp polymerase will move along, knock out these single-strand binding proteins, and synthesize new DNA on our lagging strand. Leading strand, much easier. You add the beta clamp once, it'll stay on, and you just move along. What are TER and TUS? TER are terminations to replication signals, so they are actual nucleotides in the genome. Depending on which way you're going, you'll be stopped by different TERs. So if I'm going clockwise, the first TER I can be stopped by is C. TUS is a protein that associates with TER, and only when TER and TUS are at the same location will the replisome be halted. And the idea is that you're stopping the replisome because remember, you have a replisome going in each direction, and they're going to crash into each other, and that's how you know that replication is finished. Why does E. coli replicate with such high fidelity? 4 different reasons. One, you have all 4 levels of DNTPs. So when poly3 is working, it has more or less a 25% chance to grab any one. If one was overrepresented, it would be more errors happening. But since you have an equal chance of all DNTPs, you're going to be less likely to grab you're less likely to insert the wrong one due to thermodynamics. You have 2 stages. 1, it's to base pair, and 2, if the base pair is correct, your Klenow fragment is going to be in the closed conformation and make that new DNA molecule, DNA backbone, new base. Like we said when we were looking at the Klenow fragment, that only happens if you have proper hydrogen bonds to the amino acids. So the DNTPs will try and fit in, and as soon as you have those correct hydrogen bonds with the Klenow fragment, then you close. So that's the 2-step process. 3' 5' exonuclease. So if a polymerase does make a mistake, it has the ability right then and there to cut off the incorrectly added base. And also we have a whole suite, or E. coli does, we do too, a whole suite of enzymes with the sole goal of protecting DNA. So that is another way that we can ensure DNA replication has high fidelity. With that, that's the end of this video. I will see you in the next video.